Hello students, welcome to BioBytes. For today's video, we're going to be looking at autotrophic nutrition in detail from Chapter 6, Life Processes. Now, before we go deeper into autotrophic nutrition, let's look at some basic differences between autotrophs and heterotrophs. Auto means self and hetero means other. So autotrophs literally make their own food, whereas heterotrophs have to source their food from other mechanisms. So how autotrophs manufacture their own food is they use inorganic substances like carbon dioxide and water, which is found in the atmosphere, and can prepare their own food. Heterotrophs, on the other hand, have to eat other complex organisms, even autotrophs, for their own food. When we look at the food chain, autotrophs form the primary level, whereas heterotrophs occupy secondary and tertiary levels in the food chains. Common examples of autotrophs include green plants and photosynthetic bacteria, whereas heterotrophs include a range of animals and fungi as its examples. Now we're going to discuss in detail the photosynthetic process. When we define photosynthesis, it is the process by which autotrophs use water and carbon dioxide in the presence of sunlight and chlorophyll to create oxygen and energy in the form of glucose. Basically, the reactants of photosynthesis are carbon dioxide and water. And for this reaction to occur, chlorophyll and sunlight is very essential. And the products produced in photosynthesis are mainly glucose and oxygen, and some energy is released as a byproduct as well. So if the plant has excess amounts of glucose and it doesn't want to use it, it can be stored for later use in the form of starch. So this starch can be stored in the leaf, in the stems and roots of the plant. So if we look at the steps of photosynthesis, the first step involves chlorophyll absorbing the light energy from the sun. The next step involves the light energy being converted into chemical energy, and this chemical energy is used to split the water molecule into hydrogen and oxygen. The last step of photosynthesis involves carbon dioxide being reduced into carbohydrates, and for this, redox reactions, like we have previously discussed, is commonly used. So these steps are not time constrained, meaning they do not have to happen one after the other. So most desert plants choose to absorb their carbon dioxide in the night and choose to perform their photosynthesis when the sun rises in the morning. So the reaction of photosynthesis is very, very important, and it always has to be balanced. So 6 molecules of carbon dioxide and 12 molecules of water in the presence of sunlight and chlorophyll make 1 molecule of glucose, 6 molecules of oxygen and 6 molecules of water. So this is the complete balanced equation of photosynthesis. Next, let's look at the structure of the leaf and the cross section of the leaf. So a cross section is basically if you perform a vertical cut and observe it under the microscope, what are the cellular organelles that you can observe? So when we look at the structure of a leaf, the broad flat surface is called the lamina, and the large middle vein that's running through the leaf is called the midrib. The smaller branched out veins are the smaller veins or the veinlets. So through the midrib flows 2 types of tissues. One conducts water and the other conducts food. They are the xylem and the phloem tissue, together called the vascular bundle. Now when we move on to the cross section, the topmost part of the leaf is coated by a waxy cuticle. So this keeps the leaf waterproof, and it also helps in preventing transpiration, that is water loss. So then you may observe a series of thin rectangular shapes and rectangular shaped cells at the topmost and the bottommost layer. These are the epidermal cells that form the upper and the lower epidermis. Then you may see the yellow spotted structures in the large cells. So these are the chloroplasts, and these chloroplasts contain chlorophyll, which is the site of photosynthesis. Now you may also find air spaces in between the cells. This is to allow for gas exchange, carbon dioxide and oxygen to freely flow through the leaf. At the bottommost part in the lower epidermis layer, you may observe guard cells. They form the stomatal structures in the leaf. Now we look at the experiment. Is chlorophyll necessary for photosynthesis? So for this experiment, you need a variegated plant like croton or a money plant. A variegated plant is one which has leaves which have areas that have chlorophyll and don't have chlorophyll. So the parts that have chlorophyll will look green in color, and the parts that don't have chlorophyll will look very pale yellow or white in color. These are variegated plants. Now, for the purposes of the experiment, we cannot have any previous starch made interfering with the experiment. So we place the plants in a dark room for 3 days. So when you're ready to begin the experiment, place both sets of plants in sunlight for about 6 hours. So be sure to trace the green areas of the leaf before we start boiling it in water. So after the leaf is boiled for say about 2 to 3 minutes, the next step is to immerse it in an alcohol water bath. So why do we use an alcohol water bath? It is because alcohol is a highly flammable liquid and we should never directly place it under a Bunsen flame. We should always boil it in a water bath and be safe while we do so. So you will start to observe that the leaf starts losing its green coloration and becomes pale and colorless, and the solution starts to turn green. This is because alcohol is capable of removing chlorophyll from a leaf. The last step in the experiment would now be to dip both the leaves in dilute iodine solution for a few minutes and then rinse and observe. So what can we notice while we observe these two leaves? So the areas where photosynthesis has taken place means starch would be there. So iodine is an indicator of starch. If starch is present, it would turn blue-black in color. If starch is absent, then the leaf would remain colorless. So what can we infer from this experiment? That areas containing chlorophyll photosynthesize and hence turn blue-black. Areas that don't have chlorophyll cannot perform photosynthesis, and hence this variegated leaf remains colorless. Now let's look at the structure of stomata. As previously seen in the cross section, stomata are structures which have guard cells. They are present predominantly in the lower epidermis, although some stomata are also present in the upper epidermal layers. Stomata are the site of gas exchange. They take up carbon dioxide for photosynthesis and release oxygen, which is a byproduct of photosynthesis. Apart from the stomata, different areas like the stem, roots, leaves are also performing gas exchange, but this takes place in very small quantities. So how do the stomata open and close periodically? Basically, water from the neighboring epidermal cells flow into the guard cells, and this causes them to swell up. So when they swell up, they open, and the stomatal pore or the stoma opens up, and now gas exchange is able to occur. So when the stomata needs to close up, the water from the guard cells moves back into the surrounding cells, and thus the stomata shrinks and closes up. So you might think the stomata can remain open all the time and maximum photosynthesis can take place, but this would lead to an adverse effect. If the stomata is open for a long time, water loss in the form of transpiration can occur. So the plant might lose excessive water. It is for this reason that in very hot days when there is excessive sunlight, the stomata close up to preserve its water. Now we will move on to the second experiment. We are going to test if carbon dioxide is necessary for photosynthesis. For this experiment, it is preferred that you take 2 potted plants and keep them in the dark for 3 days to get rid of all the starch that was previously present. Now take one of the potted plants and place them near a beaker containing potassium hydroxide. Potassium hydroxide is capable of removing the carbon dioxide from the surrounding area. So it will absorb any carbon dioxide which is present near the plant. Then transfer both plants under a bell jar and seal them, make it airtight using Vaseline. So this should not let any atmospheric air enter the jar. Now place both the plants with the jars in sunlight for about 2 hours. Now we're going to test for starch like how we performed in our previous experiment using iodine, following the same steps. Now what are we going to observe? The plant that was placed with potassium hydroxide would not have had any carbon dioxide to perform photosynthesis. So it would not have produced any starch. So when tested with iodine, it is going to give a colorless result. Whereas the plants that had carbon dioxide was able to photosynthesis under the sunlight and thus would have accumulated starch in its leaves. They will give a blue-black color. So this proves that carbon dioxide is indeed a requirement for photosynthesis. With that, students, we've come to the end of the video. Please like, comment below if you would like any other topics to be discussed, and don't forget to subscribe to BioBytes.